Hello everyone, welcome to my presentation on the cellular architecture of evolving neuroinflammatory lesions and multiple sclerosis pathology. This was written by Petra Kokanja and other authors. So let's get into it. So first, in order to make sense of what we're going to talk about today, we need to understand what is multiple sclerosis. So multiple sclerosis is an autoimmune disorder that ultimately attacks the myelin sheaths. Now, the exact pathology for this is very unclear, hence the study that we're covering today. Um, the next thing we need to talk about is what are myelin sheaths. So myelin sheaths are like kind of like insulation around the neurons that speeds up the nerve impulses heading up and down the central nervous system and whatnot. Without these myelin sheaths, um, your nerve impulses would be very slow, and it would take forever to respond to stimuli, and it would be almost impossible to maintain homeostatic conditions within the body. So what is meant by an autoimmune disorder? Well, an autoimmune disorder is a disorder in which your immune system, which is supposed to protect you from pathogens and maybe harmful cells and whatnot, is actually it's actually going to attack healthy tissue, which is obviously that's a bad thing, right? We don't want to kill our healthy tissue. So how does the immune system affect myelin sheaths? Well, again, that is not very clear, hence the study today. But it's pretty much well known that it has something to do with inflammation, right? And these T cells and B cells and all the a lot of the cells of the immune system, they're going to go ahead and attack glial tissue and myelin sheaths and other insulating and important tissue to the central nervous system. So, what this research paper aims to address. This research paper aims to address or elucidate the evolution of multiple sclerosis. So the problem with multiple sclerosis is that we're always taking samples from dead people. That kind of sounds pretty gloomy, but it's the truth. You know, we're not seeing the whole picture because we're not watching multiple sclerosis as it progresses from the beginning of the disease pathology to the end. So we're not getting the full picture. We're just getting the end product. And if we want any hope of developing drugs in the future, we need to know the entire pathology, right? So as we touched on before, there's inflammation in the central nervous system, and obviously inflammation is caused by the immune system because there's a flooding in of white blood cells and monocytes and macrophages and whatnots, antibodies. And these areas of inflammation are called lesions. So the other thing this paper aims to address is that lesion progression, or that area of inflammation, how it progresses as multiple sclerosis takes its course. And the last thing that this paper aims to address is how the different stages of these lesions, um, um, not only not only how they occur as a result of cell interactions, but how they affect other cells around them, right? Because we want to figure out how they're affecting the cells around them, how the cells around them are affecting the lesions, and that way maybe we can find a target to stop these lesions from progressing, or maybe stop them from occurring at all. So moving on. So the approach they took is, obviously you can't just like take a human and inject them with chemicals and induce multiple sclerosis. That would be completely unethical. So what they did is they took a bunch of rats and they induced something called experimental autoimmune disease. It's very similar, practically identical to multiple sclerosis, and this is due to the high amount of genetic information that rats share with humans. I think it's upwards of 90%. Um, don't take me, don't quote me on that, but I'm pretty sure from my biotech class it's upwards of 90%. Um, so by inducing this disease in rats, which are so similar to humans, we can make observations of the disease as it progresses in rats, and from these observations apply certain principles or stuff that we learned from the disease progression to humans. So the next probably the most important thing of the entire approach is the spatial transcriptomics. So spatial transcriptomics, what it is that Well, first of all, let's talk about observing gene expression normally. When you observe gene expression normally, you remove the DNA or the RNA from the tissue. You run PCR, whatnot, and you get these bands in gel electrophoresis, and there you go. You can see what genes are being expressed in the tissue. Now, the issue with that is you can't see exactly where these genes are um, being expressed, and and the goal of this study is to figure out how these lesions progress as a result of their cellular environment. So what we want to do is figure out exactly where these genes are being expressed so we can identify what cells are in these areas of inflammation, right? So that's where spatial transcriptomics comes in. It allows us to take a tissue sample and see where in the tissue sample that these genes are being expressed, and that way we can tell what cells are there, what cell to cell interactions they might be having, and whatnot. Okay, but the other issue with this is that the resolution of spatial transcriptomics typically isn't good enough to see single cell expression. Like we typically can't see what genes are being expressed by a single cell. We can tell where it's coming out of an area. So let's say a certain gene is being expressed, we can see where in an area that gene is being expressed, but we can't see exactly which cell in that area is expressing the gene. So this is where our next step of the approach comes in. So there's something called the Carta NA kit, which we won't get into too much detail because it kind of takes away from the paper. But basically, it just enhances our resolution so we can see down to the single cellular level, so we can see exactly which cell is expressing certain genes, and then we can tell exactly which cells are present in these lesion areas, right? And so what this allows us to do is map the exact cell-to-cell interactions that are occurring in these lesion areas. We can see exactly which cells are there, and how they might be interacting. And the last thing I want to talk about for the approach is we're doing this study in rats, right? With EAE. So we're doing the study in rats. So how do we apply this to humans? Well, obviously we need to be able to apply it to humans in order to develop drug. Like we love the rats. We don't really care about the rats that much, right? We want to be able to apply this information to humans. So what the scientists did is they took cadaver samples, like tissue samples from cadavers of the central nervous system, and they compared those the lesions. Well, first of all, these cadavers had died from multiple sclerosis, so they compared the lesions of those cadavers to the lesions that occurred in mice, and found that the progression of these lesions was very similar, and therefore we could apply the principles of the observations we made in the mice or the rats to the humans, right? Okay, so major findings of this paper. The major finding of this paper is that we now have a greater understanding of the natural history of disease. And what do we mean by natural history of disease? We don't mean some scientists who discovered it in the 1800s. By natural history of disease, what we're talking about is the progression of the disease as it takes its toll on the organism, right? So how the disease starts, how it goes through its stages, and then how it ultimately ends when the organism either recovers from it or dies, right? Okay, so we have a greater understanding of natural history of disease. And in order to have that, we had to have a greater understanding of the lesion progression, the actual area of inflammation that is eventually causing these glial cells and myelin sheaths to die, right? So what occurred in these lesions, right, for us to understand them better? Well, we found that there's actually different compartments of the lesion as it progresses, and we'll see that in a later picture, and we'll talk about it. But what happens is it starts out as a perilesion compartment. So there's this little area of cells, typically a lot of monocytes. So a lot of immune cells, a lot of white blood cells, right? And then that region of the lesion expands, and then there's an inner region called the lesion rim, and then that expands even more. There's a lesion core compartment, and then there's a late lesion stage. And we'll see pictures of all those, and we'll cover them in more depth in a second. Okay, so the next thing is that the lesions tend to form in specific areas, in specific regions of the central nervous system. It seems like a letter got cut off there, or maybe I just mistyped it, but lesions tend to form in specific regions of the central nervous system as the disease progresses. And this is very important because what it tells us is that there's cues for these lesions to form in certain areas, right? If there were no cues, if there were no biomarkers, making these lesions form in certain areas, they would just form anywhere. So what this implies is that there's some sort of biomarker or cues from the cells in that area, something they might be secreting, expressing, receptors, something. There's something there that we might be able to target to prevent the formation of these lesions in the first place, or at least stop them from progressing into the later stages of the disease. And then the last thing we want to talk about is that obviously the EAE in mice correlated very well. We already talked about this, but it correlated very well to the MS progression in humans. So therefore, we can draw observations from the EAE progression in mice and apply it to multiple sclerosis in humans. And moving on. So here's what I want to talk about, like the lesion progression. So because it's hard to understand without pictures. So what we see here is that, I don't know if you can see my mouse, but I'll just say it just in case you can't see my mouse, is that in lesion stage 1, if we look at the top row, you see the two rows? If we look at the top row with the pictures, in lesion stage 1, what we see there is the first like incoming of white blood cells. The pink is the monoclonal blood cells, right? So like kind of like white blood cells, immune cells. So as this lesion begins to expand, and then that original part of the white blood cells, they become what's called the perilesion compartment. So they are the outermost compartment, and what forms in the middle of them is the green area right there. You'll see in lesion stage 2 is the lesion rim compartment, And again, okay, again, you'll see from the pink just below it, like the robe just below it, all the pink is the white blood cells, the immune cells. So those cells are what's causing the inflammation and eventually the die-off of glial cells or diseased glial cells and the die-off of myelin sheaths, right? Okay, so then we see in lesion stage 3 is what we have is the central compartment, of the lesion, and then we have in yellow, and then we have the rim compartment just outside the yellow, and then finally we have the pink compartment, which is the perilesion compartment. You can see it's starting to like get really dispersed now in the pink, right? And then finally in lesion stage 4, we have all this green stuff. That's just pretty much to show you like, okay, all the cells are kind of disordered now. They're all spread out. They didn't really specify too much exactly what the green was meant to be. It's just called late stage lesion compartment. Yeah. And so again, looking at this bottom row just below these lesion compartment stages, you can actually see what kind of cells are there. So the pink cells are the monoclonal bodies, or they're eventually going to turn into more advanced white blood cells. And you can see there's just a lot of them, right? I mean, they're pretty much taking up the whole middle of the picture. And that's what the issue is, that indicates inflammation, and that will eventually lead to the die-off of our healthy glial cells, or at least make them diseased, and eventually the die-off of the myelin sheath, right? Because there's nothing, again, to maintain these the rest of the central nervous system cells, because that's the job of the glial cells, to maintain the central nervous system. Okay, so another important chart in the study. I didn't put in too many charts because they're kind of like complicated, and I didn't want to waste everyone's time talking about charts the entire time. But if we look to the right, figure D, we can see at the very bottom, we have you can see MC/D, and you can see that these first four columns, the CC, I can't read the second one, the GM, the WM, the amount of MC/D, which is monoclonal bodies and their derivatives, is very, very, very low. Like these little points, the smaller the point, the less of those cells. So in healthy tissue, very, very, very low. But then we can see in the PLC, right? So that is the first stage. So that is lesion stage 1, the peripheral lesion compartment. We see there's a spike. There's a spike of these monoclonal bodies and their derivatives. And again, that's an indication of rapid inflammation. So that's an indication of these lesions forming. And then we can see that it kind of grows, and we can see that right from the cell population images. We can see that these pink cells are becoming more numerous. So then we have the the rim compartment, which has even more. There's a bigger pink circle, and then the central compartment, the lesion central compartment, which has an even bigger pink circle. And then immediately after, right? Immediately after, I can't see because my video feed is blocking this off, but I believe it's a very small pink circle, very small pink circle in the late lesion compartment. So I think that one's the LLC. I believe I can't see because my video is blocking it off, but I believe there's a very small pink circle there, the late lesion compartment, and and then we can see that if you look up a few rows back to the orange circle, that is disease microglia. We can see in healthy tissue it's very low. I mean almost non-existent, right? And then we can see like as the pink circles start to get big, so do these little orange circles. We start to see a sudden appearance of diseased microglia, and and at the very end, in the late lesion compartment, we can see a relatively big orange circle compared to the previous orange circles. There's a lot more disease microglia, and these microglia are responsible for they literally are immune cells. They are responsible for protecting the central nervous system. So we see a lot of this disease microglia. So obviously, if you have disease microglia, you might get faulty immune cells that might attack the central nervous system, or just not be functional, not protected, right? And then we can also see a similar pattern for diseased astrocytes. Like even though all the circles are iffy, you can see the last circle is like a touch big as well, right? Okay, so I just wanted to point that out because I thought it was a little bit interesting. The other thing that's interesting is the T cells, right? The T cells, we look at the very bottom row, the very bottom row. I mean, those circles are practically invisible. They are so tiny, right? There's almost no T cells in healthy tissue. I mean, there are some, of course, because there's always going to be T cells, but there's very few. And then you can see that in these perilesion compartments, um, the lesion rim compartment, etc., these yellow circles got a lot bigger, right? Again, indicating inflammation, which will eventually lead to the die-off of the glial tissue and the myelin sheaths and oligodendrocytes and all of the above, right? So very bad for the central nervous system to have these sorts of cellular neighborhoods, right? We don't want these kind of cellular neighborhoods. We want all these first four columns, like this kind of balance, right? Okay, moving on. So what this means for the future of MS. So for the future of MS, so up until now, very little is known about MS. So we don't really know exactly what causes this inflammation. There hasn't been too much research on it, so that's the reason for this whole paper in the first place. So of course, so now we can do more research on the specific disease phases, because up until now we didn't really know uh, the specific phases of these lesions up until now. So this is a major finding in this paper, is that now we know that these lesions, they have specific phases, and perhaps with more studying we can figure out what biomarkers lead to the progression of these phases, and then we can of course attack those biomarkers with drugs, or just make them targets of certain drugs and whatnot. We also found that these lesions had regional preferences. So there was another very complicated chart that I didn't include here, but it shows that the lesions will begin either on the ventral or the dorsal side. And remember, lesions they're not stationary. It's not like once the inflammation starts at one point it stays. As the lesion progresses, it tends to move from either the ventral side to the dorsal side, depending on the location, or towards the middle, again depending on the location. But these lesions have preferences for regions within the central nervous system, and this was found in the spinal cord and in the brain. This was not specific to any part of the central nervous system. So what that tells us, what that must imply, is that there must be certain biomarkers, there must be certain things like sort of attracting the formation of these lesions, right? Because otherwise, why wouldn't they just form anywhere? Again, so that implies that we might be able to target something in these cellular neighborhoods, these cellular structures, right? The last thing I want to go ahead and talk about is inflammation ultimately plays a role in the onset of disease glial tissue. Of course, I mean, we've talked about this pretty much throughout the entire presentation. If there's inflammation, there's an influx of white blood cells. This isn't too new. We pretty much know that. The paper just kind of helped confirm this even more. I mean, obviously there's inflammation, there's influx of white blood cells, immune cells, whatever, and they're going to start attacking stuff. Now, in multiple sclerosis, for whatever reason, they do tend to attack the glial tissue and eventually the myelin sheaths and oligodendrocytes and whatnot. Obviously, not healthy at all. So, thank you so much. That was the entirety of my presentation. I hope it was clear and it wasn't too concise. Hopefully, that's not too much of a problem. But thank you.